Hello everyone, welcome back to Prep Pro Papers. We were discussing about TIFR-JGEEBILS 2024 previous year question. Now we'll move to the biology part of this question paper. The first question here says, a Dachshund is a type of dog with short stature. Beagles are normal size. The progeny of a Dachshund beagle mating are all short stature. Half of the puppies that are produced with the beagle Dachshund's progeny when mated with beagle have short legs, the other half are normal size. What does this tell you about short stature? Okay, we'll go from the statement to statement. So the observation that here says that all the progeny of Dachshund beagle mating are short stature. So this suggests that the short stature is a dominant trait. Let's see what confirmatory statement it says ahead. When these offspring are mated with beagles, which are normal size, half of the puppies have short legs and the other are normal size. This segregation pattern is consistent with the Mendelian inheritance, uh, where a dominant trait, which here is a short stature, will show up in the F1 progeny, as here mentioned. Now we'll check the F2 generation. The F2 generation shows 1:1 segregation of the character when the F when crossed with the homozygous recessive, that is the normal size beagle. So the correct option here is option number D. Moving to the next question. Which of the following options regarding an mRNA formed by bacterial transcription is incorrect? The transcription start site occurs a few bases before the codon. So this statement seems yes, because we know for the prokaryotic transcription, let me draw this here. So for the prokaryotic transcription, there is an up element here, and let's consider that this is our RNA start site, which is vitamin +1, and at the -10 region we have the TA which from where the transcription initiates. At the -35 region we have TTGACA, where the RNA polymerase binds. And I've already said you want the up element which interacts with the CTT domain of the polymerase enzyme. Let's go back to the question. Have a look first. So the transcription start site occurs a few bases before the start codon. This is true. The first 3 bases of the mRNA have to be 5' to 3'. This is not necessarily true that the first 3 bases to be, or the mRNA should be AUG. It can be any other mRNA also, which can serve as a start codon purpose. The transcription start site occurs a few bases after the start codon. No, it's not after, it's before. The last three bases of the mRNA always corresponded to the one of the three spot codons. No, this is invalid because at the very last basis we have the 3' UTR region of the mRNA. So the correct option is option number A. The next question is very easy. The number of alleles possible for a gene is infinite. Next we have, how does an adaptation such as better running speed evolve with natural selection? So natural selection directly produces better adaptation over time, gradually leading to the better running speed. No, we cannot say it to be directional and gradually leading to the better running speed. The natural selection makes individuals with favorable traits such as a better running speed more attractive, ensuring they produce more favorable. No, it may be a characteristic for the survival of the better, but it's not the characteristic to define natural selection. Natural selection involves random mutations, some of which are beneficial adaptation. Heritable beneficial adaptation lead to the offspring with highest level. So we have the key words here, such as the random mutations, beneficial adaptations, and heritable character leading to the highest survival. As this statement, see, in any chances, there will be some options which are more favorable, but we need to check upon the keywords which are more given in a suitable answer, the suiting our answer approach more better. Natural selection produces individuals with favorable traits such as a better running speed who survive better. Individuals with unfavorable traits do not survive, ensuring that all individuals in future population have better running speed. Again, I can say that there is always a confusion between A, B, C, D, but as C is having more precise to the definition of natural selection, we will select option number C. Now the question says, despite the large number of unique mutations described in the DIA region that causes familial hypercholesterolemia, certain mutations are found in high frequency specific population. Example, the 9.5 kb 3' deletion is found in 35% of Finnish patients. This is an example of. So we know the differences between. This is an example of founder effect, as the founder effect establishes a subpopulation as a new colony which is drawn from a larger population, and it is more and less a result of migration. So the correct option will be founder effect. Now moving to the question number 6. In our body, in cells and muscle cells share the same DNA, which of the following is true? Although they share the same DNA, both have a different genetic code, so the protein expressed are different. No, wrong. All of them are having the same genetic code. The statement is incorrect. They are different tissues, so there is a difference in their DNA. No, again, there is no difference in the DNA. The statement is true. They differ through the genes they transcribe and translate. Yes, and the major factor for these changes happening between the two cells, say for example, skin cell and muscle cell, containing the same DNA and but giving us the different characteristics of the protein synthesis is epigenetics. The statement is true. The skin cells and muscle cells differ in how many copies of the DNA. Again, there are the same number of copies of DNA in all the cells of a same organism. Now for the next question. this again you need to be very theoretical with different analytical tools used in biology and chemistry. So let's see which of the following techniques cannot be used to quantify affinity between an enzyme and its peptide substrate. Let's see for X-ray crystallography. X-ray crystallography basically gives us the atomic resolution between a bond between an enzyme and the peptide, but it's not giving us any quantity. No, isothermal calorimetry. It gives the measure by the heat changes during molecular binding. So thus giving us the quantify affinity. Surface plasmon resonance. So surface plasmon resonance it measures changes in the refractive index near the sense surface as molecules bind. Thus, again giving us a quantifying affinity result between the enzyme and the peptide substrate. The fluorescence anisotropy, it measures changes in the rotational motion of a fluorescently labeled molecule. Then again, this molecule can be either the enzyme or the peptide substrate, thus giving us a quantified affinity. So the correct answer which cannot give us the quantified affinity is X-ray crystallography. Moving to the next question is which of the following statement is correct regarding the increasing molecular weight of the proteins due to each of the post-translation modification? It's quite simple. You just need to know which molecules are used in this different kind of epigenetic modifications. So for the methylation, the group added will be CH3. For the acetylation, it is COCH3. For phosphorylation, it is PO4. And for the myristylation, it is the 14 carbon fatty acid. So we are to check upon the molecular weight. This will be the heaviest, then this, then this, and then this. So option number A. You are carrying out an enzyme reaction in the lab and obtain Km and Vmax values. While repeating the experiment, you use only 1/10 of the enzyme. What would you observe? So, when we talk about the Km and the Vmax, the Vmax is dependent on the total enzyme concentration. Sorry, concentration, whereas the Km is the intrinsic property. Let's now look upon the option. You would get the same result because the enzyme is same and hence properties are unchanged. No, the result won't be same. However, the property is same of the Km because it is the intrinsic property of an enzyme. Vmax would change, but Km remains the same. Yes. Vmax would remain the same and Km would be 1/10 of the original value. No, this will be vice versa. Vmax will get to 110 where Km will be same. Both Vmax and Km should decrease. Again, it is wrong. So option number B. Uh, Question number 10 says, rich fishing grounds, that is those that have high species diversity in population, such as those found in northern Japan or around Iceland, occur where cold and warm ocean currents mix. This is because a fanning of cold currents bringing nutrition to the surface, supporting phytoplankton blooms, providing food for many fishes. Warm currents bring nutrition from rivers. Of cold currents carry fish. Eddies set up where the current mixes from the marine habitats for many fish. So yeah, for this question, I will say it's not necessary to read out all these options. Just to know this effect is caused by the eddies effect, and the how what is the phenomena causing the eddies effect is because the nutrients gets up well to the surface, supporting the phytoplankton, and these biomass again gives the warmer carbon content and the burning to the bottom of the ocean. So setting up of a current. So thus the option number A is correct. So researchers passed three different states of E. coli, black squares, WT, red circles, and blue triangles are the mutants. See, these are the mutants. This is the wild strain. For several days, gradually increasing concentration of the antibiotic cefotaxime N is equal to 96 replicated populations per strain. Some populations went extinct during the experiment. This plot below shows the number of surviving populations of each strain. Y-axis as a function of antibiotic x-axis. Panel A shows the data from the strains carrying one copy. This is for the one copy beta lactamase gene, and panel B shows the data for strains carrying the multiple copies. Given this result, which of the following is incorrect? A 0.25 mg cefotaximine is sufficient to kill half of the Wilson bacteria. So you see here, no, because it's not at the exactly half of the surviving thing. So this option is wrong. Adaptation to CTX is enhanced by beta lactamase gene. Yes, because the mutant of the beta lactamase genes are showing better survival. Copy in not the mutant of the beta lactamase, I'm sorry, but those with the multiple copies of the beta lactamase gene are showing better survival than with those with the one copy. Copy number of the beta lactamase changes the probability of extinction. Yes, supporting the statement of the option number B. Beta lactamase is not only the factor affecting the population survival. Yes, because in these three of the mutants, in both the cases, the survival rates are different. So this is not only supported by only the beta lactamase, but also several factors acting upon. So the correct option showing that which is the incorrect option is here, option number A. The mitochondrial F-type ATPase is a remarkable protein machine in which a subunit rotates during H plus transport and one ATP is generated for 120 degrees of revolution. ATP hydrolysis releases 50 kilojoule per mole and the transport of H plus costs about 19 kilojoule per mole, while the exact number depends on the cell condition. Approximately how many proteins need to cross the membrane to achieve a full revolution? So Let's consider for a full revolution, which comprises of 360-degree, and 1 ATP is generated for 120 degrees. So there will be 3 ATP generated. Now for 3 ATP generated, ATP hydrolysis is releasing 50 kilojoule per molar. So the energy released for the 3 ATP will be 3 into 50, that is 150 kilojoule per mole. And the transport it requires for the hydrolysis of ATPs is 19 kilojoule per mole because of the transport of the H plus. So the H plus concentration here will be equal to 150 upon 90, which will be roughly approx 10.89. So the closest option to this is option number D. A peptide has the following sequence: ATHRMN, which is given here, ADHMARAMEN. At pH 3.6, the net charge of the peptide is likely to be. So you need to know which abbreviation of the amino acids it is, which amino acid, and their pK values. So let's consider. I'm having a little doubt in this question also, because I feel the answer given in answer 2 is not right. Let's see. Because let's consider this is the N terminal and this is the C terminal of the protein. Now for the pK values, here pK value is 9. T will not T will not giving any ions. H will give us 6. Again M and A will not give any charge. R will have the pK 12. Again AM will not have any give us any charge. E will give us 4, and N will give us, N will have a value around 2 to 3. So we'll see which are protonated and which are deprotonated. This will give us at pH 3.7, this will give a plus charge, this will give us a plus charge, this will again give us a plus charge, this will give us minus, and this will give us a minus charge. So the net total comes to be around +1. If any of you knows the correct way to solve or is any better approach, because answer they have provided is +2, but I feel +1 is the correct option, please comment below. Move to the next question. A neuron makes 3 synapses on muscle so as to produce a sudden movement. However, each synapse is unreliable and only conveys a signal with a probability of 0.5. Successful signal propagation by any one of the 3 synapses will result in a muscle contraction. What is the probability that muscle will contact when neuron fires? Okay. So there are 3 synapses. So we'll make a combination of 3. Say these are the 3 synapses. Every time only one conveys a signal. So we'll see how many times the conversion signal will happen. If this conveys and these are shut up, and then if these are on, this is off, then these are on. The combination can be this is off, this is on, and again this is off, then this is off, this is on, this one is off, this is off, this is off, and this one is on, this is on, this is off, and this one is off. So we see that the total combination here is 1, 2, 3, 4, 5, 7, 8. So 8 is the total number of events happening, and what all are the successful because only the one conveys. So this one gives a signal, this one, this one, this one, this one, this one. is 7. So the option is 7 by 8. And now next, move to the next question. Last question of this previous year question of 2024. In an vitro transcription experiment, the transcription and both the strands of DNA were separated and analyzed for the base composition. The results obtained is shown in the following table. Determine which DNA strand is serving as the template for mRNA synthesis. So for G pairs with C, we're looking upon right now for the mRNA. A pairs with T. There is no thymine in the mRNA. C pairs with G and U pairs with A. So there are 24 G in the mRNA, so there should be approximately 24 C, which is here in strand 2, and then there is 29.6 A in the mRNA, so there should be approximately 30 T here. For C, there is 20 G, 20 C, so there should be a 20 G value, which is again in the strand 2. For U, there should be approximately 25.8, that is approximately 26 A, so that is again in the strand 2. So the correct option is strand 2. I hope you are liking our videos. So keep looking upon more. You need to subscribe to our channel Prep Pro Papers, and we'll meet you up in our next scene. Thank you.